Kia ora team, welcome back to the 2.5 Genetic Variation and Change 4-credit external video series. This is video 13. In this video, we're diving into the two key processes that drive evolution, natural selection and mutation. By the end of this lesson, you should be able to explain how natural selection changes allele frequencies in a population's gene pool, describe how mutations introduce new alleles into the gene pool, and discuss examples of both natural selection and mutations in action. In the previous lesson about population genetics, you learned these definitions as well as how to calculate the frequency of any given allele in a gene pool. You also learned that biologists use allele frequencies to measure genetic change in a gene pool over time to study how populations are evolving. Over the next two videos, you will learn about the 4 evolutionary processes that cause changes to the allele frequencies in a gene pool. This video focuses on the first two, natural selection and mutation. Populations normally produce more offspring than what the environmental resources can maintain, so individuals have to compete with each other to survive. For example, takahe have to compete with other grazing mammals for alpine tussock grasses for food, and takahe have to compete with each other for mates. There are also plenty of non-resource related environmental factors that individuals have to be well adapted to survive, such as the climate and temperature, predators and disease. Takahe, for example, live in harsh alpine conditions and have to move to forested areas when snow covers the mountains. Takahe also can't fly and are therefore at the mercy of ground predators such as stoats. Natural selection is the process by which individuals with alleles better suited to their environment survive and reproduce more successfully than others. The beneficial alleles of these reproductively successful individuals are more likely to be passed on to the next generation. If the allele continues to be beneficial, it will increase in allele frequency in the gene pool over time. So the environmental factors I mentioned earlier, both biotic factors such as competition and predators, and abiotic factors such as the climate, act as selection pressures on these alleles. When environmental factors change, different alleles could be selected for. Beneficial alleles tend to increase in frequency in the gene pool, while harmful alleles tend to decrease in frequency in the gene pool. Harmful alleles could be entirely lost from the population if it is selected against due to lower chances of survival and reproduction. In this example here, the trait we're focusing on is this moth's ability to tolerate frost conditions. The big A allele gives the moth the ability to tolerate frost, allowing the moth to survive once the frost arrives. The little a allele makes the moth more vulnerable to the frost, and as a result, none of the homozygote recessives survive the frost. So, natural selection acts on gene pools by reducing the reproductive success of individuals with phenotypes that are poorly suited to the environmental conditions. As we can see, the little a alleles become less common in the gene pool because its allele frequency decreased from 43% before the frost to 29% after the frost. And on the flip side, natural selection also acts on gene pools by increasing the survival and reproductive success of individuals with phenotypes well suited to the environmental conditions. As we can see, the big A alleles become more common in the gene pool because its allele frequency increased from 57% to 71% after the frost event happened. Let's look at a real example. In New Zealand, there are around 21 different species of stick insects. They come in all shapes, colors, and sizes to camouflage against their surroundings. In this case, the bird predators act as the selection pressure. Stick insects with alleles that lead to better camouflage survive and reproduce more successfully than others. The beneficial alleles of these reproductively successful stick insects are more likely to be passed on to the next generation. If the allele continues to create good camouflage, it will increase in allele frequency in the gene pool over time. So, how do new traits come about? Through mutations. A mutation is a random, permanent change in the base sequence of a gene, giving rise to a new allele in the gene pool. In fact, mutations are the only source of brand new alleles, and are therefore the ultimate source of genetic variation. If a mutation occurs in the gametes, then that mutation will enter the gene pool and become subject to environmental selection pressures during the process of natural selection. Mutations actually occur very frequently, and most mutations have little to no effect. Some mutations are harmful, and very rarely a mutation produces a new allele that's beneficial. In the graph above, we can see the frequency of 3 alleles over time. Originally, there were only 2 alleles, big A and little a. In the beginning, the environment favored the big A allele, and we see a steady rise in its allele frequency over many generations. The little a allele was probably harmful, so over time we see its decline in allele frequency. Then, in the 7th generation, a gametic mutation occurred and a brand new allele entered the gene pool, A'. In the 13th generation, there was a change in environmental conditions. There were new selection pressures that favored the brand new mutant allele, giving it a competitive advantage. And the mutant allele steadily increased in frequency over time and becomes established in the gene pool. When you look at this picture, what do you see? Or more importantly, what don't you see? Originally, all peppered moths were the pale speckled type because their gene pool only contained one type of allele, the speckled allele. So you could say that the speckled allele at that point was fixed because it made up 100% of the gene pool. But around the year 1819, a gametic mutation occurred in one moth. This mutation produced a new allele that gave the moth a much darker, almost black appearance. At first, the dark color allele was very rare in the population. Since the environment at that time in 1819 favored pale moths, the dark allele didn't spread much in the gene pool. It was actually a disadvantage because the dark moths stood out on the background of lichen-covered trees. Then, the Industrial Revolution happened and dramatically changed the environment. The poor air quality killed the lichen, and the trees became covered in soot. Suddenly, the dark-colored alleles became more useful. The selection pressures have changed. The birds ate the pale moths, while the rare dark moths survived and reproduced. Within just a few decades, this once rare mutation spread widely throughout the gene pool. This peppered moth example shows mutation in natural selection in action. Mutation created the new dark color allele, and on its own the mutation was neither beneficial nor harmful. Its value depended entirely on the environment, and when the environment changed, natural selection favored the dark allele, so its frequency increased in the gene pool over time. Here's another classic example. Sickle cell disease is one of the first clear demonstrations of natural selection in humans. I've taken all of these illustrations from an 18-minute long video on HHMI Biointeractive. It's really interesting, so I've put the link in the description below so you can watch it. The sickle cell allele, HBS, entered the gene pool through a gametic mutation in the gene for hemoglobin. This is the protein that carries oxygen in red blood cells. Normally, the hemoglobin A allele makes round, flexible red blood cells that can move easily through blood vessels. But the sickle cell mutation causes hemoglobin to clump together, changing red blood cells into stiff sickle shapes. Individuals who are homozygous for the sickle cell allele have sickle cell disease. Their sickle-shaped red blood cells can block blood vessels and cut off oxygen supply to tissues and organs. This causes intense pain and organ damage, and it ultimately shortens their life expectancy. Because of this, people who are homozygous and have 2 copies of the sickle cell allele are much less likely to survive to adulthood and reproduce. So on its own, the sickle cell allele seems really harmful. And because the phenotype it causes is so harmful, we would have expected the allele frequency to decrease in the gene pool. So why is it that today there's around 7.8 million people with 2 copies of the allele living with this disease? The answer is in natural selection. In regions where malaria is common, such as parts of Africa, the Middle East, and South Asia, the normal hemoglobin allele can actually be a disadvantage, and the abnormal sickle cell allele can have beneficial effects. Malaria is an infectious disease caused by the single-celled Plasmodium parasite spread by mosquitoes. These parasites infect red blood cells, feed on their hemoglobin, and use the red blood cell to reproduce. If untreated, malaria can destroy huge numbers of red blood cells, cause anemia, organ failure, and even death. So in areas with malaria, individuals who are homozygous and have 2 copies of the normal hemoglobin allele are more likely to die young and not survive to a reproductive age. Interestingly, the HbA and the HbS alleles are co-dominant, meaning that the heterozygotes who possess both alleles in their DNA produce both types of red blood cells, the normal red blood cells with normal hemoglobin and the sickle-shaped red blood cells. These heterozygous individuals don't usually suffer the full effects of sickle cell disease because they produce enough normal hemoglobin, and they also produce enough abnormal hemoglobin to make it harder for malaria parasites to survive and reproduce. Remember that the Plasmodium parasites feed on hemoglobin, but the abnormal hemoglobin in sickle cells is not in the form that parasites can feed from. This gives the heterozygote individual resistance to malaria. So in regions with malaria, heterozygous individuals are more likely to survive and reproduce compared to homozygous individuals. The homozygous individual with no copies of the sickle cell allele would likely die of malaria, while the homozygous individual with no copies of the normal hemoglobin allele would likely die of sickle cell disease. This is why we see high frequencies of sickle cell allele in areas where malaria is common. Do you see the overlap between the purple regions, where the allele frequency of the sickle cell allele is high, and the orange regions where malaria is prevalent? These are in the African regions, Middle East, and South Asia. So in areas with malaria, the selection pressure of malaria favors heterozygotes, keeping the sickle cell allele in the population despite the serious health problems it causes in homozygotes. This is an example where natural selection maintains 2 alleles in the population because each has an advantage under certain conditions. But in parts of the world without malaria, the sickle cell allele is purely harmful. Without the selection pressure of malaria, the sickle cell allele is gradually lost from the population. Because homozygous individuals with 2 sickle cell alleles and no normal hemoglobin, they die young, and heterozygotes no longer have a survival advantage. This is a clear example of natural selection and the ability of environmental selection pressures to influence the allele frequencies and gene pools. That's the end of this lesson, so by now you should be able to explain how natural selection changes allele frequencies in a population's gene pool, describe how mutations introduce new alleles, and discuss examples of both natural selection and mutations in action. Thanks for watching, and I'll see you in the next video.